Okay, so I'd like also to thank the organizer for this great day. I learned a lot, so coming by train in an hour, and you have great speakers in the field. So thanks a lot for the invitation and for giving me the opportunity to present some of our work. And so, yeah, we're going to talk about P53 reactivation therapy. So I would like to start with some kind of a provocative statement. So we've learned a lot about P53, and as we heard today, there's great work ongoing in the field. Today, there's more than 70,000 publications, I think, on P53, which makes it one of the most studied, if not the most studied molecules in biomedical science. The bad news, I think, is that to some extent, very few, if any, patients have benefited from this work directly. And I think that the field is very much in need of a breakthrough in the field that will help translate some of this work into the clinic and turn really P53 into a cancer killer. So, so my lab is very much interested in this concept of P53 reactivation therapy, which has been introduced several years ago by Karen Vosden and David Lane, Arnie as well, contributed to this idea, which is a very simple idea, and posited that in tumors that retain wild-type P53, the pathway is inactivated anyway by a number of mechanisms. And of course, in the end, if we understand those mechanisms, we might be able eventually to pharmacologically intervene and reactivate dormant P53, which is latent, functional, but inactivated in these cells. Of course, this is not as easy as this cartoon depicts. There are numbers of challenges associated with P53 reactivation therapy, and one of the challenges is try to identify the right target. I think there's a great deal of discussions in the field which target needs to be targeted in order to get P53 optimal P53 reactivation. And eventually, once you identify the right target, what you need is, of course, develop drugs that pharmacologically eventually inactivate those targets. And the challenges there are really to identify targets that specifically would activate a full P53 response, hopefully an apoptotic response, so inducing cell death. And of course, importantly, we've heard today that P53 activation in normal tissues can be very detrimental, and ideally these drugs would specifically target the tumor cells and leave the non-tumor cells unharmed. And finally, once this is achieved, the next step is, of course, to identify the right patients and the right tumor types to be treated. And of course, eventually, what we need is also a number of biomarkers that will help stratify patients that will and predict whether or not patients would respond to this. In the context of P53 reactivation therapy, one, of course, would like to focus mainly on tumors in which the frequency of P53 mutation is very low, such as melanoma. We have decided to focus on melanoma simply because P53 is mainly wild type in these tumors in more than 90% of the cases. So melanoma is very interesting for that aspect, but also it's a very important clinical challenge in the field, and moreover, it's an interesting paradigm to study the ins and outs of targeted therapy. There is a new drug that have been introduced recently in the clinic, and that gives very beautiful anti-tumor responses. And I'm sure you've all heard about this. It's based on the fact that malignant melanoma are addicted to the MAP kinase signaling pathway. So the MAP kinase signaling pathway is active in virtually all melanoma. This is a consequence of BRAF mutations in about 50% of the cases, and in about 30-35% of the cases due to mutations in NRAS. Now, it's been known for many years that if you inactivate this pathway in melanoma, you're going to induce decreased proliferation and possibly cell death. And so this pathway really represents one of the Achilles heels of melanoma. And this has been recently explored in the clinic by developing drugs that selectively target the activated form, the mutated form of BRAF. So again, present in about 50% of the cases. So one patient out of two that get this mutation can be treated with drugs such as PLX4032. And as you can see on these images, you get massive anti-tumor responses. This is one patient before treatment, metastatic melanoma, dissemination in many different tissues and organs. Only 2 weeks after treatment with this specific inhibitor, you get massive anti-tumor responses. Unfortunately, these responses are only transient, and a few months later, these patients come back to the clinic with a disease which is no longer responsive to the treatment. So it's quite exciting, but of course, there's still a long way to go. There's a number of needs. We definitely need to identify molecules which eventually will affect the viability and the proliferation of cells that acquire resistance to the BRAF inhibitor. And importantly, we all think in the field that combining drugs that will target different signaling pathways, possibly, will synergize eventually with a BRAF inhibitor in promoting killing of the BRAF mutant cells. And to come back to the analogy of Achilles' heel, it seems that one arrow is not enough. What we think is that we need to target these lesions with numbers of arrows that target multiple signaling pathways. So this was for the background. Now let's go back to the idea of P53 reactivation therapy and what about the right targets. So I have to say that virtually all pharmaceutical companies that have been thinking about this project all focused on MDM2 as a possible potential candidate. And MDM2, as you've heard today, is an important negative regulator of P53. It promotes its degradation and inactivates its function. So as you probably all know now, Lubo Vasilev at Roche developed a molecule which is known as Nutlin, which binds specifically MDM2 and physically disrupt the interaction between P53 and MDM2, allowing P53 reactivation. Subsequently, a number of molecules which have similar properties have been developed by many different companies, and they are known as MDM2 antagonists. So from our view, I think that the MDM2 antagonists have at least two major issues. And the first one is toxicity. So we and others in the field have inactivated MDM2, so genetically ablated MDM2 in numbers of tissues in mice, for example, in postmitotic neurons, in red blood cells, in the heart. This is work from Gigi Lozano's lab. And we have inactivated MDM2 in smooth muscle cells of the GI tract. So these cells are terminally differentiated. We inactivate MDM2 in these cells. What we see invariably is upregulation of P53, induction of cell death, eventually cellular dilation of the intestine and death of the animals. So in other words, our work, as well as the work of others like Jeff Wall, more recently Gerard Evans, clearly indicates that MDM2 plays an important role in controlling adult tissue homeostasis by maintaining P53 levels low. So of course, this raises serious concerns about the use of MDM2 antagonists in the clinic to some extent. Another issue that was actually discussed already a bit this morning when Laurent discussed about MDM2 is that many cancer cells are relatively immune to MDM2 inhibitors. Most cancer cells exposed to Nutlin, for instance, activate a P53 response, but these cells undergo mainly cell cycle arrest. Very few of those activate the apoptotic program. So again, there may be a number of explanations for this. We discussed about MDM2 recruitment to chromatin this morning. We think that there are also a number of feedback loops that are activated upon P53 activation that could, of course, also attenuate the P53 response. But we also think that one major player that contribute to the decreased sensitivity of cancer cells to MDM2 inhibitors is MDM4. So we have heard about MDM4 or MDMX, so we're talking about the same proteins for the people that are not really expert in the field. We have provided recently, well, actually over the years, evidence that MDM4 is overexpressed in many different human cancer cells, and that MDM4 overexpression attenuates the response to MDM2 specific inhibitors. So a few words about MDM4. Actually, I was not going to show this, but since PGP is here, I'm going to show this because this is work done in his lab, and I would like to thank him publicly for allowing me to do these experiments in the lab, because I think that they were quite important at the time. So we knocked out MDM4 in the mouse and showed that this leads to an embryonic lethal phenotype, which is completely rescued on a P53 deficient background. These experiments put really MDM4 on the map at the time. And similar experiments were carried out in Gigi Lozano's lab, by the way. So clearly highlighting an important role for MDM4 in controlling P53 function, at least during embryonic development. Subsequently, we have developed much more sophisticated mouse models. I will not go into any of the data, but summarize some of our findings with this cartoon. And this is, again, also summarizing our work, but also the work of Jeff Wall and Frank contributed to this, and many others. So the idea is that we think that at least during embryonic development in some tissues, the two proteins, MDM2 and MDM4, contributes to P3 regulation in a relatively independent way, to some extent at least. We have provided clear genetic evidence that MDM4 is able to contribute to inactivation of P3 function independently of MDM2. It actually binds P53 in its transactivation domain and to some extent decrease its ability to transactivate target genes. So now this provides a rationale for why, indeed, in tumor cells that express very high levels of MDM4, inactivating MDM2 might not be sufficient to get the full P53 response. One of the major differences between MDM2 and MDM4 is in adult tissues. So what we have done is also inactivate MDM4 specifically in numbers of tissues in the adults. And the conclusions is that in some cases we see slight P3 activation, but by and large we see almost no toxicity, meaning that tissues are just fine, in adult tissues without MDM4 are just fine by and large. So for example, we have knocked out MDM4 in the smooth muscle cells of the GI tract, as opposed to loss of MDM2. This leads to absolutely no phenotype. Mice are totally fine. We have done this in numbers of tissues and confirmed that this is the case. So in contrast to MDM2, MDM4 is by and large dispensable for adult tissue homeostasis. And in fact, we now have finally, it took us many, many years to have good antibodies to detect endogenous MDM4 levels in adult tissues. And we find that, in fact, MDM4 is expressed at high levels in embryonic tissues, to some extent in the brain, but in most adult tissues, actually, MDM4 is actually not expressed, or expressed extremely low levels. In contrast, we have provided, again, evidence that MDM4 expression gets upregulated in cancer by numbers of ways. And I will show you our work, our recent work on melanoma. We have provided evidence recently that MDM4 overexpression in melanoma is one of the key mechanisms that contribute to P3 activation. And this is work from Agnieszka Gambarska in the lab, and Flavie Luciani contributed also to this work. So I'll go very quickly through this set of data because this was published about 2 years ago now. And again, because of great quality antibodies that finally put our hands on, we were able to show that MDM4 protein is indeed expressed at very high levels in about 65% of human melanoma. These are 40 different primary tumor samples, so from different stages of melanoma development. So these are very early lesions. These are nodal, so regional dermal metastases, nodal metastases, and distant metastases. What's interesting is that MDM4 upregulation seems to be occurring even very early during melanoma genesis. These are very early lesions, and again, about 60–65% of these lesions have already very high levels of MDM4. What's interesting is that normal melanocytes do not express MDM4, as well as benign nevi also do not express. So it's really very specifically upregulated in the tumor cells. We could confirm this by immunohistochemistry, which allows us to look at many, many more samples, and we confirmed this over more than 200 samples now, that indeed MDM4 is expressed at high levels in the nuclei of 65% of cutaneous melanoma. So to establish a causative link between overexpression and melanoma genesis, we use mouse models. This is published again. So we've developed a mouse transgenic for MDM4, which is a conditional transgenic mouse allele, which allow us to turn on MDM4 expression only in specific tissue. Here we turn on MDM4 only in melanocyte using a tyrosinase CRE allele, and we combine this with the tyrosinase NRAS allele, which leads to melanoma. So this is the human allele, which is found in about 35% of human melanoma. So these mice express constitutively the NRAS in all of the melanocyte. They are very pigmented. Eventually, these mice develop melanoma, but it takes a very long time. If you inactivate P53 function in this, on this background by removing P53, you get acceleration and you get similar results if you overexpress MDM4. So MDM4 overexpression, just like loss of P53, cooperate with NRAS in promoting melanoma genesis on this background. We have also data which are not published, and I don't have time to go into, indicating that MDM4 can also cooperate with BRAF in promoting melanoma genesis in mice. But the real question in the context of cancer therapy is, are tumor cells, melanoma cells in this case, which are already established, dependent on MDM4 to grow and proliferate and eventually to survive? So to address these questions, we use a number of model systems. We have developed a number of short-term culture melanoma lines, which indeed express, or many of them express MDM4, and knock down MDM4 using an shRNA-based approach. This is before, after knockdown, before, after knockdown, before, after knockdown, numbers of lines, and by and large, the conclusion is very dramatic and very consistent. Decrease in MDM4 expression in these cells leads to a very dramatic decrease in the ability of the cells to grow, as, for example, scored by colony assays. And importantly, in some cases, we see a very clear induction of an apoptotic response, as measured here by Caspase-3 7-glow assays. This increase in apoptosis is p53 dependent. By knocking down p53 on this background, we decrease the induction of apoptosis to some extent. I should also mention that concomitant to the increase in apoptosis, there is a dramatic cell cycle arrest in many of these cultures. And to some extent, we could not rescue this by decreasing p53 function in these cells. So it seemed to be a p53 independent component also on the oncogenic functions of MDM4 in melanoma. But by and large, this data indicates that MDM4 protects melanoma cells from p53-induced growth inhibition and to some extent from p53-induced apoptosis. So this is in vitro. We have done a number of experiments also in vivo. This is one of them. We basically use a xenotron translation. So these are xenografts in which we give, so these mice are given doxycycline. They were infected before with a virus allowing expression of an hairpin that targets MDM4. So if you give the doxycycline to mice, you turn on the hairpin, you decrease MDM4, and then you can measure tumor growth, and then you can see that there is dramatic impact on tumor growth. In fact, there is tumor regression in this model. So we think that, at least in the context of melanoma, MDM4 might be a right target to some extent, because MDM4 is not expressed in most adult tissues. It's expressed at high levels in a large number of cases, and knocking down MDM4 leads to activation of p53. So in the context of p53 reactivation therapy, it might be an interesting target. So how do you target MDM4? Unfortunately, nuclein and small molecules that target MDM2 do not efficiently block the interaction between MDM4 and p53, and there is no small molecules that inhibits this interaction as yet. But what we used at that time is a staple peptide. So these are peptides that are chemically modified. They are locked in alpha helical configurations. These peptides were designed by Federico Bernal when he was at the Dana-Farber several years ago. And he has a number of those peptides that specifically bind either MDM2 or MDM4 to some extent, and that binds both with similar affinities. We use a peptide which binds MDM4 with a greater affinity than MDM2. So this peptide binds MDM4 with a 40-fold increased affinity as opposed to MDM2. So we expose the numbers of melanoma lines to increasing concentration of the staple peptides. These are wild-type melanoma cultures, and you can see that there is a dramatic decrease in cell viability, again, by increasing the concentration of this peptide. Melanoma cells that are mutant for p53 do not really respond to this. They are rare, but they do exist. And importantly, in these cultures, which are p53 wild type and sensitive to the drug, if you knock down p53, you decrease to some extent this effect, and you decrease the increase in apoptosis that we see also when the cells are exposed to this drug. So these peptides, which are known as, which is known as SAH-p53-8, induce p53-dependent apoptosis in human melanoma cells in vitro. Remarkably, this also works to some extent in vivo. So again, xenograft experiments, we now inject IV this peptide for 5 consecutive days and follow tumor growth over time. This is the growth of a tumor which is on a mouse that is injected with a control peptide, and we see that we have stabilization of the disease when we treat the mice with this peptide that targets the MDM4 p53 interaction, indicating that at least to some in the in vivo context, targeting the MDM4 p53 interaction prevents or decreases melanoma growth in vivo. Now, the most exciting, I think, observation was that this staple peptide that targets the interaction again between MDM4 and p53 sensitized cells to the BRAF inhibitor. So what we've done is taken numbers of culture that have BRAF mutations, expose these cultures to BRAF inhibitor alone, so to basically EC50 doses of these inhibitors, and to the peptide alone. And when we combine the two, we have a dramatic synergistic effect. In fact, we can measure the combination index, and it's all cases lower than 1, indicating that there is strong synergies. So these data are exciting in a way because they indicate that combining BRAF inhibitor with agents that target MDM4 might allow or lead to longer lasting anti-tumor responses to some extent, or even possibly to eradication of the disease. We also have data, I don't have time to show you, that cells that acquire resistance to the BRAF inhibitor remain sensitive to MDM4 targeting, which is also interesting. So some of the main messages from this type of work, from this part of the work, is that MDM4 protein, I didn't have time to show you, but we will come back to this, but not mRNA, is overexpressed in 65% of human melanoma. That overexpression of MDM4 in the mouse cooperated with NRAS and with BRAF. And that MDM4 protects from P53-induced apoptosis and growth inhibition. And one of the main messages, at least to us in the field, is that despite P53 being wild type in these tumors, we have the impression, based on this data, that P53 function is to some extent compromised in human melanoma, and this is by and large, or to a great extent, due to MDM4 overexpression. This has implications. So if we manage to target MDM4, we get a reactivation of P53, and we can dramatically sensitize the cells to BRAF inhibitors, as well as induce killing of cells that acquire resistance to BRAF inhibitors. Now, this staple peptide, I should mention that there is a company in the US, Leuven Therapeutics, who are developing these molecules and actually now going into phase 1 and phase 2 clinical trials. With a peptide that targets both MDM2 and MDM4. And we can discuss this, what are the advantages and disadvantages of targeting both. We are also hoping to convince them to go also with some peptides that target specifically MDM4, at least to some extent. Now, again, as I said, there's no small molecules. I think there are numbers of big pharmaceutical companies interested in trying to develop small molecules that target this interaction, but so far nothing really came out of these studies. So we decided to actually take a step back and think about another way to target the pathway. And we thought that if we were to understand the mechanisms that contribute to MDM4 upregulation of melanoma, we might be able to target this event as opposed to the direct physical interaction between P53. And so this is work done in collaboration with Ernesto Guccione in Singapore, and this is work done by Michael and Karen in the lab. So the experiment started by a quite interesting observation, an intriguing observation, which is indeed that if you take a number of short-term culture lines, you have cells that express very high levels and cells that do not express any detectable levels of MDM4. But if you look at RNA levels, this is an RNA-seq experiment looking at total RNA in these cells. We have basically virtually no differences between the RNA levels in these cells. So basically, in other words, cells that express, who do not express MDM4 still express high levels of RNA. So I guess I have Robin's attention now, so this uncoupling between RNA and protein. We also looked at the promoter activity using ChIP-seq experiments. We've done H3K27 acetylation ChIP-seq on all of the melanoma lines that we have, and we find that there is a very beautiful peak in front of the MDM4 locus, indicating the promoter of MDM4 is on in all of these cultures. So the promoter is on in these cultures, the RNA is made, but in some cases you get expression, and in others you don't. So then we went back and looked in silico at data from TCGA. So the TCGA has more than 300 human RNA-seq data for human melanomas, and we looked at the RNA-seq reads for MDM4 and found that there are actually two main transcripts which are found in melanoma, the MDM4 full length, the coding transcript, but there's also another transcript which is found in a relatively high numbers of these patient samples, is MDM4S. And this transcript is well, quite known in the field. It's a transcript that corresponds to, which is actually depicted here. It basically simply lacks exon 6. So by lacking exon 6, this introduces a stop codon in exon 7, and this leads to a premature stop codon, which eventually would predict that this RNA would undergo non-self mediated decay because you get a stop codon which is far away from the poly A signal. Elegant work from Frank recently showing in the mice that if you force expression of the MDM4S, you get a null phenotype, you actually don't get any expression of the MDM4S. So basically, the conclusions so far from the experiments that we have done, and I will show you some more later, is that MDM4S is a transcript which is likely to be relatively unstable, and if the protein is made, it will be truncated because you have stop codon, and we know now that the protein is actually very unstable as well. So now going back to the TCGA data, the 300 samples, we looked at the ratio between the full length and the short in the different samples, and we find, surprisingly, that, well, interestingly, that in about 65% of the cases, the full length is predominant over the S, and in about 35% of the cases, it's the opposite. The short form is predominant compared to the long form. So we've designed a number of tools to look at those two transcripts. So a very simple one is a qPCR-based assay with 2 different primers, and we can detect in one single PCR reaction both transcripts, the full length, the longer form, and the short form. And then by looking at the expression and the ratio between these 2 transcripts in the numbers of human melanoma cell lines, we indeed find that they have dramatic differences, as you can see here. In some cultures, you get mainly expression of the full length, and in some cases, you get mainly expression of the short form. So the ratio between the full length and the short varies dramatically from samples to samples. And now if you look and superimpose these 2 protein levels, you have a very beautiful fit between the ratio in the full length, short, and the presence of MDM4 in these cells. So if you have a full length ratio high, you get expression. As soon as you get a full length ratio which is becoming lower, then you get no expression. Okay. So we have looked at this splicing regulation in numbers of cellular and systems and organs in which we know that the MDM4 levels are varying and modulated. And one of them is going back to the Western blot I showed you earlier, looking at MDM4 levels in adult tissues. So we have detection, we can detect easily MDM4 protein in embryonic and in the brain. And again, the full length to short ratio is very high in these samples, whereas in the other human adult tissues, these samples, the ratio is very low. Another cellular system which is very attractive is the mouse embryonic ES cells. We know that these cells express high levels of MDM4, but if you now expose these cells to retinic acids, you induce differentiation. This leads to a decrease in MDM4 levels, and again, this correlates with a dramatic shift in the full length to S ratio. Now we're going back to our melanoma samples. So again, we looked at the numbers of short-term culture lines, and again, you have dramatic differences between the ratios, and this correlates very well with the presence of the protein. These are clinical samples, these are short-term culture lines. I also would like to mention that we now, by looking at our TCGA data and by drawing artificially the line at 65%, because we know that 65% of the human samples express MDM4, we can basically obtain a ratio over which we would predict that the MDM4 protein would be expressed. And now we can go back to in silico analyze many different tumors and look at first of all, we have to confirm that the prediction is correct, and we have done this, and clearly when the ratio basically is a log2 ratio, which is over minus 0.58, and then you get basically expression, and then there's a very, very nice fit there. So what I'm trying to tell you basically is that in embryos, you need MDM4 expression to buffer P53 function, and this is achieved by promoting exon 6 inclusion, and this leads to MDM4 expression, and then you get expression of the MDM4 protein. Now, in most adult tissues, there is a splicing switch which occurs, and basically you make mainly the MDM4S, which leads to no protein, and that's why in most adult tissues there's no expression of the MDM4. Now, tumor cells reactivate some kind of an embryonic program, so basically reactivate the alternative splicing switch, promoting exon 6 inclusion and promoting expression of the MDM4. Now, I don't know what you all know about splicing. I learned recently about this by, of course, based on this data. So the idea is that, of course, you have a core splicing machinery, but you have also a number of splicing enhancers and silencers, which are able to bind specific sequences, which are either intronic or exonic. And these splicing enhancers or silencers can really, in a specific way, regulate splicing of different genes. And so we have performed a siRNA screen that targets all of the splicing enhancers known to date for looking at splicing enhancers that affect this specific event. This is work done by Ernesto in Singapore. And we found that SRSF3 is one of the key regulator of MDM4 exon 6 inclusion. There is a lot of data suggesting or confirming this possibility. Just show you this experiment. So if you knock down SRSF3 by using 5 different hairpin RNA, looking at, again, at the ratio between the full end and the short by PCR, you could see that knocking down SRSF3 leads to a dramatic decrease in expression of the full length over the S. Now, there's more to the story I don't have really time to go into, but what's known about SRSF3 is that it's an oncogenic splicing enhancer, it's a MYC target. It's regulated by phosphorylation, and among which AKT-mediated phosphorylation promotes its activity. So there's good evidence to link SRSF3 increased activity and MDM4 exon 6 inclusion in cancer. Now, can we use this therapeutically? What we've done, the answer is potentially yes. So what we've done is design a number of antisense oligonucleotides that bind the exon-intron boundaries of exon 6. And by doing so, we might be able, and actually we'll show you that we are able to promote exon 6 skipping in human cancer cells. So we have the different designs. We use morpholinos and antisense oligonucleotides. We are now working mainly with morpholinos, but it also works quite well with just classical antisense oligonucleotides. So to show you that this works quite well, so these are 2 human melanoma cultures and cell lines. So we transfect, in this case, the morpholino in the cell. And again, we're looking at the ratio between the full length and the S, and you get a dramatic decrease, again, of the full length to the short. This leads to a dramatic decrease in MDM4 protein abundance, activation of P53 response, and then we get the classical responses, meaning that there is the decreased colony formation, increased sensitivity to BRAF inhibitors, and so on and so forth. So all this to say that we think that it might be possible now to target the pathway by really regulating specifically this splicing switch, and we have provided evidence, I didn't have time to show you, that if you expose cells that acquire resistance to BRAF inhibitor, the cells become sensitive to this MDM4 specific antisense oligonucleotides, and again, these antisense oligonucleotides synergize with BRAF inhibitors. So is this clinically relevant, really? Can we use antisense oligonucleotides in the clinic? I've been surprised when talking to people at ISIS and different companies that are really working into trying to revive the idea of using oligonucleotide in the clinic. In fact, ISIS has 40 ongoing clinical trials right now. And so basically, indeed, there is a new rush in the field, the use of antisense oligonucleotides, and simply because the chemistry has evolved very dramatically, and we can use those now in vivo. They're very stable in vivo, and it seems to attract a lot of attention in the field right now. And so we've performed experiments in vivo to show that indeed the system works. So we have developed at KU Leuven a number of human melanoma PDTX models. So we basically take a piece of a tumor, transplant this in the mice, expand, and then eventually treat these mice. So these are 2 cohorts of mice treated with a control oligonucleotide. Tumors grow. If you treat with the oligonucleotide that target MDM4 splicing, you reduce dramatically tumor growth. And I was very impressed by this. So these are 6 mice treated with the control oligonucleotides, 6 mice treated with the oligo targeting the MDM4 exon 6 splicing. Look at again the PCR. Basically, you get almost a complete shift in expression of the different isoforms. Basically, you promote the switch from the full length to the short in this. This leads to a dramatic decrease in MDM4 expression levels, a decrease in Ki67, so proliferation, increase in apoptosis. So it works in vivo. So I think it's quite exciting in a way, so we could target the pathway using a different approach. Now, I have also data to show you, but maybe I'm going to run out of time now, so I'm going to skip this, but simply to show that we can combine in vivo as well BRAF inhibitors with the azos and show that you get much better responses than when you treat the mice with the BRAF inhibitor alone. And also I'd like to point out, and I briefly alluded to this idea that MDM4 possesses some P53 independent oncogenic function. Of course, these functions are likely to be non-affected by drugs that target the P53 MDM4 interaction, but by targeting MDM4 protein abundance, now we can basically impact on both aspects of MDM4 function, its ability to suppress P53 function, as well as to inactivate, or to function oncogenically independently of P53. So, quite excited about this. Now, is this a melanoma-specific thing? Well, I told you that we can now in silico predict whether or not MDM4 will be expressed based on our measurement of the ratio full and short. Well, we actually think that the enhanced exon 6, MDM4 exon 6 inclusion is a widespread oncogenic mechanism that leads to upregulation of MDM4 in many different cancers, and that this event is targetable using this type of antisense oligonucleotides. We have done experiments now to show that using this antisense oligonucleotide, we can also decrease viability and growth of breast cancer cells, for example, that express high levels of MDM4, as well as, for example, neuroblastoma cells. So the proof of concept is on melanoma, but this applies to other tumor types. And so this is my last slide, or before last. What I just told you today is that in the context of P53 reactivation therapy, and at least in the context of melanoma, we think that MDM4 might be a good and a safe target, much safer than MDM2, for instance. That staple peptides are in the clinic right now, and we hope that maybe we'll bring antisense oligonucleotides to that stage as well. And importantly, what we also would like to mention here is that we by no means claim that we should use these drugs as single agents, but we should combine them, especially in the context of melanoma, with MAP kinase targeting agents, for instance. Also, the last point I want to make is biomarkers. So, of course, for targeting MDM4 and looking at patients and making so stratifying the patients, what we think is that our simple PCR method that detect the full length to S ratio and predict whether or not the protein will be expressed might be a useful tool to discriminate the patients, as opposed to just look at the protein levels, which is much more complicated in a clinical setting. And with that, I'd like to stop and thank you for your attention. These are the people that contributed to the work. So, I mentioned them as I discussed the work. I just would like to really stress that Ernesto Guccione was a very important player in the MDM4 splicing story, and these are the collaborators of the previous paper on MDM4 and melanoma. So thank you for your attention.